Okay, well, let's go ahead and get started here for today. So you don't have to turn in your problem set now, but if you want to, you can. Or just after class instead of having everyone run down here. But I decided to delay the problem set due date because there are people that really need to leave campus and go to, Denver or Longmont or wherever. So, okay. So I delayed that. I also made an announcement that I'm going to delay quiz 10. Obviously, that's not for traveling reasons. It's an online quiz. But I wasn't entirely sure where I was going to get to today in today's lecture, so I just decided let's just push it off. I think that we'll get to a place in today's lecture that you could probably go ahead and take it. But just to, you can wait until Friday, okay? Make sure that you get it done on Friday, though, okay? The last thing I want to say, I also posted, and I said I would do this, that video of the electron transport chain that I showed you, at least portion of that, the complex 1. So that's been posted in Lecture 9 module in Canvas. So take a look at that. There's like 7 minutes of material in that. It's actually really good. So some of it we didn't really cover in this class, but it gives you a little bit more complete picture of electron transport chain. Okay? All right. So a riddle, what animal can jump higher than a 5-story building? Huh? Not a bird. No, these are jumping animals. No one knows? Superman is no. These are real. I can jump a 5-story building. I'm Captain America, but anyway. All of them. All of them. That's right. Because buildings don't jump. Right? It's a fact, right? I mean, it's a fact. Buildings don't jump. Animals do. So, animals, all of them jump higher than 5-story buildings. Yeah. Okay. I'm just going to leave it at that, okay? So that's a good snow day riddle, I guess. Not a snow day yet. It's a snow day in 60 minutes. Okay, anyway. So we left off right here on Monday. So, and we're looking at really a table that covers what we had done in our last group work. Essentially calculating how much ATP can be generated from a single glucose oxidation event, okay, 1 molecule of glucose. This does, though, assume that NADH, the PO ratios of NADH equals 2.5 ATP and FADH2 equals 1.5 ATP, which I said that is also going to assume a C ring of 9 subunits. So we may or may not get to the C ring today. We'll find out. Okay, certainly we'll get to it on Friday if we don't get to it today. Okay. All right, now, one more thing to say here in regards to the electron transport chain, and then we'll step fully into discussing ATP synthase and its mechanism, okay? But I wanted to go ahead and talk a little bit about uncoupling. I've got another question for you as well, okay? So, but uncoupling, we have said that these two processes are coupled, meaning that they're codependent on one another. There are compounds that have been discovered that will lead to the uncoupling of them. And by uncoupling, we simply mean that the electron transport chain can run independently of the ATP synthase apparatus. Okay? You can't do it vice versa. Okay? You can't uncouple ATP synthase from an electron transport chain. ATP synthase can't really run and make ATP unless there's a proton gradient. Okay? So that can't be established without the electron transport chain. However, electron transport chain can run in the presence of what we call an uncoupler without a functional ATP synthase. And the uncouplers will essentially do this. They will take protons from here, grab onto them, and move them directly across the membrane in some fashion. Okay, and that would be, that red circle would be an quote-unquote uncoupler. We'll look at a couple here in a little bit. Uncoupler. Okay. Now let me go ahead and ask a question, okay, on this. This will lead to some interesting discussions as well here. Okay, but if uncoupling is occurring, what would happen to the PO ratio in the mitochondria? Okay. So what would happen to PO ratios in a mitochondria that has been exposed to an uncoupler? What do you think? Are they going to go up? Are they going to go down? Talk to your neighbor about that and why that would be the case. And your marks, get set, go. Okay? All right. About 30 seconds left. Okay, 10 seconds left, so submit an answer if you haven't done so. Says that I have 151 responses. I'm skeptical. I don't think there's 150 of you. I think that counter must be wrong, okay? All right, go, okay. Increase, decrease, it would go down. Okay, it would go down, it would go down, I don't know, increase, lower, decrease, the PO ratio would decrease. Okay, lower. Okay, so let's look at our word cloud here. Okay, decrease. It looks like the majority of you, based off of that, would say decrease. Okay, so that is indeed correct. So let me come back to this slide here. Okay, what we essentially see with an uncoupler is something that's going to take protons that are being pumped to the p side and then going to move them across the membrane independent of something that can make ATP. Right? So we talked about the fact that NADH will lead to the translocation of 10 protons. Right? They go here, here, and here. Those are going to be pumped from electrons derived from NADH. Those 10 protons, if it takes 4 protons to make 1 ATP, then they can lead to the production of 2.5 ATP. Let's say, though, however, that our uncoupler grabs 4 of those protons and moves them independent of the ATP synthase molecule. Well, that then is going to lead to less protons that can make or generate ATP. So your PO ratio is like NADH, instead of making 2.5 ATP, you can make 1.5 ATP, or even less, depending on how much uncoupler is present. Okay. Now, those protons that cross the membrane, okay, and are released on the inside, that energy is going to be released as heat. Okay. Now, uncouplers, let's go ahead and look at how they might do this. So there is what we consider unnatural uncoupling, and then there would be natural uncoupling. Let's talk about unnatural uncoupling. So you are looking at 2 different molecules that have been discovered that are unnatural uncouplers. Let me go ahead and highlight the top one. The bottom one does essentially the same thing, but let's just look at the top one. It's a simpler molecule. 2,4-dinitrophenol, or just simply known as DNP, is an uncoupler. And you'll see here that it's got a proton bound to it. This would be something that you would expect to happen at lower pHs. Okay, it would be in a protonated state. If it were to get to a place where there would be higher pH, it could deprotonate at higher pH. Okay. Higher pHs are seen in the matrix, and lower pHs are seen in the intermembrane space. okay, especially at the surfaces of the membrane, which is where this would be found. DMP, and I don't know if we know this, I don't know this, but I don't know if even the scientific community knows this. DMP can pick up the proton, for sure, we know that. How it gets through the inner membrane is, I don't think, known. Okay, my suspicion is that it would have to be taken by some transporter. Okay, so there must be some transporter that has some specificity for it, interestingly. And then it will get into the matrix and it will release that proton. And then, of course, there will be some heat released. Now, interestingly, that molecule, DMP, was discovered as like the wonder drug in the I think it was 30s that it hit the market. And it was a wonder drug because you could lose weight using it without doing any work, right? So it was taken off of the market because it was killing people. Right? You don't want to really tinker with the electron transport chain and oxidative phosphorylation in this way. So, and one of the pathologies of the individuals that were taking this was hyperthermia. They had increased body core temperature, and that can actually be very dangerous, as you might understand and know. All right, so DMP, actually you can still find, if you Google DMP, you can still find people that are selling it and pushing it off as this weight loss drug. But it's especially dangerous because there are some bodybuilders that have heard about this, and it's a good way to lose fat, and so they want to lose fat and be all ripped and everything. I don't take DMP, if you're wondering, but okay. Some of you caught that little subtle remark. Some of you are like, yeah, I can tell that you don't take DMP. Anyway, you're certainly not ripped. Anyway, so it's those kind of things. But anyway, so don't take DMP. Okay, so that's our moral of today. Don't take DMP, it can kill you. Yeah?
Is it like, if it's still being used, is it like if you take it in a really low concentration, it can be effective at what it's supposed to do without causing...
Without causing death, right? There are other symptoms that you would have. The question is can it still be effective at lower concentrations? And yes, it can be. The problem is dosing it. How do you dose it? Okay, because everyone's metabolism is different. And the medical community and certainly the FDA is not going to allow experimentations on a drug that we know kills people. Okay, especially for what is trying to be treated. Okay, there are other ways that are just as effective at losing weight. Okay, so that this will never be approved as an FDA approved drug. Okay, FCCP would be something similar. Okay, it has a similar modality. Okay, now that would be unnatural uncoupling. Don't take DMP. There is natural uncoupling. Okay, natural uncoupling can be found in brown adipose tissue. So brown adipose tissue, I'm going to highlight what this would do in certainly a human being. Brown adipose tissue is enriched in infants in particular. So the thing about an infant is that they have a really high surface area to volume ratio, meaning that they have a very difficult time regulating internal body temperature, right? Because they lose a lot of heat for a very small amount of volume. Okay, and so they have quite a bit of brown adipose tissue that is surrounding their vital organs. Now, brown adipose tissue, the difference between brown adipose tissue and what would be called white adipose tissue, aside from the color, is that in brown adipose tissue, there's a lot of mitochondria, and that's why they're actually brown, because there's significantly more mitochondria in that tissue. Okay. Now, what you are looking at is just a portion of a pathway that can lead to what's called non-shivering thermogenesis. Brown adipose tissue, the reason that it's encasing these vital organs is to bring and regulate body temperature to keep vital organs at the appropriate functioning temperature. Okay? And how that's done is, first of all, through just the hypothalamus sensing that the body is cooling down. Temperatures are going down. That will lead to a series of signals that will eventually lead to the release of norepinephrine. Norepinephrine will bind to a beta-adrenergic receptor, the G protein coupled receptor. Okay, very simple, very similar to the epinephrine beta-adrenergic receptor we talked about. That will lead to, of course, activation of G protein. G protein leads to activation of adenylyl cyclase. Adenylyl cyclase makes cyclic AMP. Cyclic AMP activates protein kinase A, which we've talked about. Okay, all of that we've talked about. What we haven't yet talked about, and we're going to go ahead and talk about this when we get to lipids, but PKA will increase what's called beta oxidation, and the rate of essentially fatty acid oxidation. So lead to the increase of triacylglyceride breakdown to fatty acids. Fatty acids will then be converted to carbon dioxide and water, and a whole lot of NADH and FADH2 is made in that process. Okay? That's going to feed the electron transport chain, as we've talked about. That's going to lead to, of course, translocation of these protons. Okay? Now, why am I telling you all this? Well, I get to this here. That is called, UCP1 is called the uncoupling protein. Okay? UCP, uncoupling protein. That moves these protons back through this membrane and produces heat. Okay? So if body core temperature starts to go down, specifically in an infant, which has lots of brown adipose tissue, these signals will essentially lead to the production of heat through non-shivering pathways. Shivering is another way to do it, right? Just produce a bunch of kinetic energy, right? Burn ATP and produce heat. Okay. This case, though, is uncoupling protein uses the proton gradient that would make ATP to make heat. This is one of a family of proteins, uncoupling proteins. Okay. You'll also find these proteins in brown adipose tissue in like hibernating animals and other interesting physiological things. Okay. So, in fact, I think the first uncoupling protein that was discovered was named thermogenin, okay, for its role in producing thermal energy. All right, so that's uncoupling. DMP, unnatural, don't take it. Okay, brown adipose tissue, you don't have much of it. Okay, so questions? Any questions on this? Yeah.
If there's some kind of signaling defect in the production of the UCP1, like in the transcription of the gene, in translation, I guess, in tissues where it's not supposed to be present, is that like lethal, or is that a disease state?
Oh, I don't think so. Right. So what you're saying is like an epigenetic change that would lead to increased expression of uncoupling protein. Is that a disease state that's been observed? I don't think so. I would have to check on this because I don't know the answer to this question, but I think that the uncoupling protein may be a gene found in mitochondrial DNA. And so regulation of that expression, I'm not sure of. Although, having said that, I'm not sure if that would be true either, because your normal mitochondria don't express the uncoupling protein, and there's not as sophisticated methods of shutting down genetic expression in mitochondrial DNA. It's pretty small. So it's probably a genomic gene, and I don't know if it's ever been observed to be expressed ubiquitously. If it were, it would be problematic, yes. Yeah. Other questions? No? Okay. I think that then is going to do it in terms of electron transport chain. Of course, today we've talked about inhibition of that electron transport chain and then a coupling of the oxidative phosphorylation. What we want to do for the rest of lecture 9, we certainly aren't going to finish this today. We'll finish it on Friday, though, probably. Will be to talk about the structure and mechanism of ATP synthase. Okay, we've now worked very hard to find out how a proton gradient is established. We understand that the proton gradient can lead to ATP and a certain amount of ATP based off the number of protons that are flowing across that membrane. So what we want to do then is to look at how ATP synthase can make ATP using a proton gradient. Okay, and ultimately this is going to come down to conformational changes in a protein. However, before we do that, we want to look at, and to do that, we need to really understand the structure of ATP synthase and its thermodynamics. Okay, so what you were looking at here would be a slide. We have one slide on the thermodynamics of ATP synthase. And if you understand this slide, you'll understand lots of what we're going to talk about on Friday, okay? But let me do this first. So let's talk about a typical enzyme, and we could even say typical enzyme like, let's say, phosphoglycerate kinase. And all of you said, ah, yes, phosphoglycerate kinase. Isn't that an enzyme in glycolysis, catalyzes the 7th step in a substrate level phosphorylation event? You got it. You guys are really on it. People that can make it through a snowstorm like you did, I'm sure, know that, okay? So, but anyway, phosphoglycerate kinase, we could talk about others. This figure is from your book. I'm not really sure why they drew it like this, but I'm going to go ahead and get rid of some things here. And I'm going to add some things to this, okay? So I'm going to come over here, and we're going to draw essentially the reaction coordinate diagram of this enzyme. And we'll just say E plus S. The S in this case, substrate would be ADP and 1,3-bisphosphoglycerate, going back to the 7th step in glycolysis, okay? And so the very first step, okay, of an enzyme, hopefully you know this is, of course, binding its substrate, so that would make the ES complex, okay? Now, in a typical, what we call substrate level phosphorylation event, the next step to make ATP is going to be where the energy is needed. Okay, and that's what's shown here, this large hill. Okay, and that will lead to EP, where P is ATP and 3-phosphoglycerate. Okay, ATP being what I want to highlight. They have, get rid of this, they have P here. EP meaning the product is still bound to the enzyme. Okay. The last step in the enzyme will be just simply a release step. Okay, E plus P. Now it might be more steps than that, but this will suffice. Okay. And what I want to highlight for us here is that this step where all this energy has to be used, this would be energy to make ATP. And in substrate level phosphorylation, that energy is going to be provided by the other high energy compound, 1,3-bisphosphoglycerate in this case. Okay, we'll provide that energy is simply dephosphorylated and gives this phosphate group up into ADP. Okay. Now, move over here and look at the thermodynamics of ATP synthase. So that's the molecule of interest in oxidative phosphorylation. Okay, and what we have here, okay, coming back to this would be E plus S. Okay, the enzyme plus a substrate, ADP, and inorganic phosphate. Okay. There is, of course, the binding event that will make ES, nothing really different here than your regular enzyme. Okay. Now, here's where the major difference comes, because the next step, this one here, the small activation energy, is going to make EP, where P is just ATP. There's nothing else. Okay? It doesn't have 1,3-bisphosphoglycerate. It doesn't have one of these other high energy compounds that it can drive the phosphate from and make ATP. You'll notice also that going from ES, and S, by the way, is, well, they have it listed here. I don't need to draw it. ADP and inorganic phosphate, that's all bound to the enzyme. It's actually a small decrease in free energy, indicating that the production of ATP and phosphorylating ADP is actually energetically favorable. Okay, which is pretty cool. Okay, now you'll notice though that the next portion here, okay, there has to be this absorption of energy to then go from EP to E plus P. And we can say that this is the energy to release, not make, but release ATP. And our key difference here is release versus make, right, between our two enzymes. Turns out that ATP synthase has almost a picomolar KD for ATP. Really strong affinity, okay, for ATP. It forms the number of non-covalent interactions with ATP, all of which have to be broken to get ATP out into solution where it can be used. Okay. To break those non-covalent interactions, the binding energy between the enzyme and the product requires significant amount of energy. And so what we are going to be looking at then in the production of ATP by ATP synthase is essentially a molecular crowbar that can remove ATP from the active site of this enzyme. Okay. Not some high energy compound like what we'd see in substrate level phosphorylation to make ATP. Okay. Now if you understood that and understood what I just said, then everything else from there is downhill. Okay. And some of you are saying it's never downhill in biochemistry. Okay. It's less steep of a grade than normal. Okay. Questions? No? Okay, awesome. Let's go find our crowbar and how that crowbar can be moved by a proton gradient. That's what we're doing. Okay, so let's look at the structure then of ATP synthase. Okay, overview first of all. On your left, I mean, the difference between what you're looking at on your left and on your right is just, you know, obviously a drawing, a cartoon rendering of it, an artist's rendition of this. And what you see on your right is high resolution structure. We know the structure of most of ATP synthase, but not all of it. Okay. Now, we will oftentimes, or this enzyme is oftentimes referred to as F1. That's not an I, F1. Got to get rid of that. There we go. F1 ATP synthase. Okay. This looks like a 0. It should be an O. I'm going to go ahead and make it a little bit more pronounced O. It is O. It's not 0. Okay. O stands for ligomycin. Ligomycin is an inhibitor of ATP synthase. Okay. All right. But when we call it FO, F1, FO, there's two really main components of the enzyme. And I'm going to go ahead and circle what's called FO. All the subunits here that have or have been named with a letter from the English alphabet are part of FO. So that's FO. Everything outside of that with Greek lettering for its subunits is F1. Okay. Both of them are needed for the enzyme to make ATP, as you might imagine. There are a large number of subunits listed here. We're going to go through, not all of them certainly, but we're going to talk about the main highlights. Okay. What you see on the right is missing some subunits. We don't have the crystal structure of all of the subunits. Okay. If you were, though, to carry this out, the membrane would be right here. Okay. So that's the membrane. FO contains what would be considered the transmembrane region of the enzyme. Okay. F1 is in the inside. Okay. Inside here. Now, let's go ahead and go and look at F1 component first, and then we'll look at FO component. So on our next slide, what we're going to do is essentially look at all of this minus these two. Okay, what's in blue and pink. Right, so this is the majority of what would be considered F1. F1 as a total is 9 subunits, only 7 of which are shown here. Okay? And sometimes we'll refer to this as kind of like a lollipop structure, okay, because it looks like what you see in blue looks like a lollipop, and then there's this green extension coming out of it that looks like a stick. Okay, 7 subunits that you're looking at here make up that lollipop structure. If we put our eyeball right here, okay, we'll get this view over here. Okay, so now we're looking at it from the top. And when you look at it from the top, we have the 7 subunits listed, and I'm going to go ahead and highlight these. There are what we call 3 alpha and 3 beta subunits. Okay, And each alpha and beta subunit make up a pair. And I'm going to go ahead and circle the pairs here. Here's one of the pairs. Okay. Here's another one of the pairs of alpha and beta subunit. And then finally, here's another one of the pairs. Okay. And you can see alpha, beta, alpha, beta, alpha, beta. Okay. So they come in these pairs, 3 each, and they alternate as we go through them. Now, when you look at what's in between them, this guy, let's use, I guess, this kind of greenish color. This guy right here, that's called the gamma shaft, or sometimes it's called the central stalk. There's only one of them. It's the stick of the lollipop. If we're to look over here on the left, this portion of it here, C, you can see that entire stick. Okay. Essentially, what we're looking at here is what you look at here, but we've removed 2 of the alpha and beta subunits, so that you can see the stick extending into the middle of them. Okay. Now, the alpha and beta pairs, this is really important to understand this whole thing. Okay. Each alpha and beta pair comprise an active site for ATP production. Essentially, the active site is sandwiched between the alpha and beta subunits. More in beta than it is in alpha, but you have to have components from each of them. Each alpha and beta pair has the possibility of adopting 3 different conformations, and those conformations are listed here. There's an O conformation, okay, that would be no S substrate, ADP and inorganic phosphate, or ATP. I guess, let me go ahead and write ADP, PI, ATP. It's empty, open, okay? That conformation, if you look down here, is listed right here, O, okay? This alpha and beta pair is in the O conformation. If we're to go clockwise from that, and this is important, this direction, if we're to move clockwise in the clockwise direction, the next alpha and beta pair is going to be in what's called the T conformation. And the T conformation is ATP bound. You can see ATP here. High affinity for ATP again. Okay. And then finally, the last one here, this is what we call the L conformation. And the L conformation has ADP, an inorganic phosphate bound. And that will be counterclockwise to the alpha and beta pair in the T conformation. Not counterclockwise, clockwise, I'm sorry, clockwise. Now, what dictates what is in O, what is in T, and what is in L? Well, what dictates what's in O, T, and L is the alpha and beta pair's interaction with the stick, the central stalk or the gamma shaft. Okay, and the gamma shaft, okay, is an asymmetric subunit and has different interactions with each of the alpha and beta pair that is contacting. Right, and we can just put let's just do this here. Let's just put a dot here on the central stalk or the gamma shaft and say wherever that dot's pointed, that alpha and beta pair will be in the open conformation. Okay. Now, if that then can be turned in the direction of this arrow and move that pink dot, okay, essentially the central stalk, in that direction, it will force a T to O conformational change. And if you go from T to O, you're going from ATP bound to empty, which means that ATP has been removed. Okay, so our molecular crowbar is the central stalk. If it can keep moving through that, it will just keep forcing conformational changes. So this, if that happens, this will go from T to O. Now, interestingly, this will then, the one adjacent to it that has ADP and inorganic phosphate bound, that will go to T. This will go to L. And it will just keep doing that as this thing turns in the clockwise direction. Okay. Now, again, just to highlight this, if it goes T to O, ATPs come out. Okay. Friday is a really good day to talk about what turns that central stock. Okay, so we'll look at how that's going to turn and how a proton gradient can force that. And we'll get into some details, but we can't get into a ton, okay? All right, so I will see you on Friday. Please be safe on your way home. You are now on officially a snow day. I know I'm the dirty son of a gun that brings you in right before snow day occurs, okay? All right, have a good rest of your day.